Welcome everyone to today's GlycoNet ACS collaboration. It's my pleasure to introduce a good friend of mine, Charlie Fehl, who is an assistant professor at Wayne State University. So Charlie obtained his bachelor's in biochemistry from the University of Michigan back in 2009. Prior to obtaining his PhD at the University of Kansas back in synthetic and medicinal chemistry in 2014. He then conducted postdoctoral research at the University of Oxford with Ben Davis, where I interacted with Charlie to quite an extent due to his really amazing work in O-GlcNAc, and as well as I think both chemical biology methods as well as bioinformatic methods. So Charlie then transitioned to Wayne State University in 2018, and I think since then has been really doing fantastic work in chemically controlled tools for O-GlcNAcylation. I think he'll tell us about some of that work today. So thank you so much, Charlie. We're looking forward to your talk.
So, since starting our group, we've focused pretty intently on trying to unpick some of kind of the spatial temporal aspects of O-GlcNAcylation during signaling. And so just a brief outline of the work that I'll present today. We'll go through how we kind of view O-GlcNAc-driven cellular processes, and then outline some of our objectives to track what we call rapid or fast O-GlcNAcylation events in cells. We've developed a tool we call PhotoGLcNAc. That's a light-based probe that's able to release or track O-GlcNAc sugar events, as well as an outlook. And I'll start off by thanking the people that really contributed to some of the work that I'll talk about today. Our first project is really led by a former postdoc, Jakaya, and a really stellar grad student, Courtney, in our group. Sydney, Alexis, and Gary have taken on this project as well for new applications. And Yimin and Zach have been putting together a biosensor that I think Christina is going to like, since we do a kind of similar thing and would love to pick her brain on a little bit more. And Sahid is working toward biological applications of some of these tools. And I'd also like to thank all of our group members and collaborators, because it's been a great journey so far. All right, so we see, we study a glycosylation process that occurs on intracellular targets called O-GlcNAc glycosylation. And this is the addition of a monosaccharide and acetylglucosamine on serine and threonine residues on proteins. It's a little bit different from many glycosylation reactions that are found on cell surfaces, because the two enzymes that transfer this, O-GlcNAc transferase, or OGT, and O-GlcNAc-ACE, or OGA, are both extremely promiscuous and actually have thousands of different protein substrates. So it's a monosaccharide modification rather than some of the oligosaccharides that build up on cell surfaces. The transferases and glycosidases that act on this are extremely promiscuous, and there are only a small number of them. And this happens on soluble proteins rather than membrane-anchored proteins. And so the large diversity of sites that can get O-GlcNAcylated allow this process to be involved in many different cellular functions, some of which that have been presented in this meeting and many others are proteostatic events, especially in neurodegenerative disease. So Matt Pratt's group calls this molecular Teflon for preventing alpha-synuclein for aggregating. And Suzanne Walker and David Vocadolo's groups have developed very nice inhibitors for OGT and OGA enzymes. Mike Boyce's group almost looks at the reverse of Matt's group, looking at O-GlcNAc-driven binding events, sort of a molecular Velcro in certain cases, that can act as reciprocal signaling tools versus, say, phosphosites, which can also modify serine and threonine residues. And then Ben Davis's lab and John Hanover's lab have developed some pretty excellent tools to look at the epigenetic aspects of O-GlcNAc as well. And some of these are occurring on histone complexes, and these can modulate transcription. What we like about thinking of O-GlcNAc processes is that they help allow cells to sense their environment. And so The concentration of glucose available to a cell will dictate the overall level of O-GlcNAcylation events on proteins. That's very well studied by Jerry Hart's group and some of the co-founders of the field. And Natasha Zakara's group and Xiaoyang Yang have really profiled how these modifications occur during stress responses as well. So it's a very dynamic system that can actually cycle on certain sites on the minutes time frame. Some of the different mechanisms and timescales of how O-GlcNAcylation can control cellular events can be summed up in, say, metabolic reprogramming in the transition from cells into cancer, where increased glucose flux will elevate the products of the hexosamine biosynthetic pathway, which is UDP-GlcNAc primarily, the substrate for O-GlcNAc transferase. That can affect epigenetic processes ranging from DNA methylation and demethylation. We do some work on some of the TET proteins. It can also modify histones or some of the nucleosome remodeling complexes. And it can even modify transcription behaviors either through the C-terminus of RNA polymerase or some really nice work by Suzanne Walker's group in controlling alternative mRNA splicing pathways. And this can happen very, very quickly within 30 minutes or so. What are less studied aspects of O-GlcNAcylation is that some of these events occur very rapidly within minutes. So some data from Jerry Hart's group following the particular kinase CAMK4 in cardiomyocyte tissues, when these heart cells are stimulated with ionomycin, we can see a very rapid drop in O-GlcNAcylation of a particular threonine site and then recovery within 10 minutes. At the same time, that threonine 200 gets phosphorylated and then also removed in kind of a reciprocal fashion. So kind of an interesting way that cells can control their signaling behavior. In neurons, a group has shown that OGT activity spikes about 1.5-fold upon depolarization and then returns to baseline in about 10 minutes. And certain events are also activated during growth factor signaling, such as insulin stimulation, which can also act on the 5-minute or so timescales. That being said, some O-GlcNAc sites persist for days, but my group focuses on some of these quicker aspects of O-GlcNAcylation. And so how do cells fine-tune O-GlcNAc events so quickly? One mechanism that we're particularly interested in is called nonsense-mediated mRNA decay, or NMD, whereas some of our transcripts, or many of our transcripts on our genes contain internal stop codons on certain introns. And depending on alternative mRNA splicing pathways, if premature stop codons are left on an intron, those pre-mRNA molecules are rapidly degraded and aren't turned into a functional protein. Regarding O-GlcNAc, Suzanne Walker's group published an awesome paper last year that showed that the level of O-GlcNAc in cells can dictate alternative splicing processes for the transferase for O-GlcNAc OGT or the glycosidase for GlcNAc modifications OGA, and these can happen quite quickly. So under high O-GlcNAc conditions overall in the cell, presumably certain splicing factors are modified such that OGT retains a stop codon and is rapidly degraded on the transcripts, whereas OGA is allowed to functionally express, and the reverse actually happens upon low O-GlcNAc levels being detected. OGT is expressed and OGA is degraded. So it's a nice system to maintain a pretty tight homeostasis for a cell. There's a lot of great tools to identify O-GlcNAc proteins. And some of Christina's work has captured over 2,000 glycosites in single proteomics experiments. And I think the grand total of known O-GlcNAc sites on human proteins is up around 7,000, with some work by Stephanie Olivier van Stickelen. Where we want to come in and fill some gaps in the literature, aside from those few examples of fast O-GlcNAc sites that we know are in these within half an hour time point before the OGT and OGA levels are allowed to rebalance themselves by presumably alternative splicing events, And so one of our tools we call PhotoGLcNAc. It's simply photo cages placed onto metabolites that lead to O-GlcNAc modifications. And we can either trigger GlcNAc-driven phenotypes, or we can use these to install some chemical biology tags for profiling these sites. We've also developed an intracellular proximity labeling tool based on a GLcNAc binding motif and a proximity labeling domain. These allow us to capture some of these intracellular O-GlcNAc sites that would respond to conditions. I'll talk more about the photo cage tool today and touch briefly on our proximity labeling ID strategy. So our approach here was to attach photo caging groups onto GLcNAc metabolites. And so in one instance, we just take a peracetyl GLcNAc sugar and attach a violet light cleavable photo cage called diethylaminopumerin on the anomeric position. When this goes into cells and we decage it, that can go through a few metabolic stages to become UDP-GlcNAc and glycosylate proteins. And so the way that this experiment works in practice is we'll feed our cells with our caged GLcNAc molecule for 4 to 12 hours. We'll wash away any probe that was not taken up by cells. And at this point, we put it in phenol red free media so the phenol red doesn't interfere with the violet light. And then we'll do a light pulse and then take different time points as we go along. Sorry about that. And do a Western blot to profile some of the O-GlcNAc, just the overall changes in O-GlcNAc levels. And so we see a fast spike within 5 minutes, which is essentially as fast as we can freeze the cells after our probe is released. And then we see kind of an evolving change in O-GlcNAc patterns within 2 hours, which disappears overnight. So we see this kind of fast phase and then an equilibrium phase. And so we were wondering, briefly, we synthesize these just by doing a pretty standard glycosylation reaction. We wanted to use these to actually try to activate a GLcNAc-driven phenotype. And so we turned to a paper from 2008 that had a nice phenotypic assay that follows NF-kappa B signaling as driven by O-GlcNAcylation events. And here, the P65 subunit of NF-kappa B has a threonine glycosite that's essential for its nuclear entry. And something that we found interesting in this paper is that if they stimulate with high glucose, because this was a hyperglycemia study, it takes about a day for the NF-kappa B to be coerced into going into the nucleus. Whereas if this paper stimulated with TNF-alpha, which is a canonical NF-kappa B signaling component, then their GFP NF-kappa B probe would enter the nucleus within minutes. And so because glucose is correlated with O-GlcNAcylation, but only a small subset of glucose molecules are taken onto the UDP-GlcNAc metabolite that's necessary, we hypothesized that we could specifically probe the role of O-GlcNAcylation using some of our tools. And so we transfected 293 cells with this GFP tagged NF-kappa B probe and put them in low glucose media. And we were pleased to see that compared to our negative control, which regardless of treating with light or no light remained in the cytosol, or our positive control, which regardless of light, when stimulated with TNF-alpha, migrated into the nucleus, we could controllably activate NF-kappa B nuclear translocation by incubating with our PhotoGLcNAc probes and then decaging the system with light. We wanted to take this a little bit further and identify which types of proteins, what the scope of these faster O-GlcNAc-driven events could be beyond NF-kappa B. And so we turned to a tool called, or a strategy called metabolic labeling. This is pretty well established for profiling glycoproteins in the literature. And we use a probe developed by Matt Pratt's group called 6-azid O-GlcNAc, which we really like because it's quite selective for O-GlcNAc transferase rather than other forms of GlcNAc-containing glycans. And so we hijack the cell's metabolism using the 6-azid O-GlcNAc to install an azide group on GlcNAc-containing glycoproteins. We'll take our lysates and then treat those with copper click chemistry conditions to install either fluorophores or biotin enrichment handles. In this experiment, we're just clicking on a fluorophore to show that this will, in a dose-dependent manner, label O-GlcNAc glycoproteins. And we can see that we see a lot more glycoproteins being labeled by fluorescence than we do with our O-GlcNAc Western blot, because the antibody sensitivity is less than a fluorophore tagging every GlcNAc protein in the sample. So we wanted to extend our PhotoGLcNAc probes with some of these chemical biology tags like azides. And so again, there are quite a few great tools that have been designed for this for different purposes, but we chose 6-azid O-GlcNAc because it's quite selective for OGT, as reported by Matt Pratt, and Shi-Chen has used it pretty selectively as well. So Jagaya installed some of these photo caging groups on 6-azid O-GlcNAc, and the experimental condition was similar. We would feed cells with 6-azid O-GlcNAc caged probes, waft them away and label. And we got kind of good news and bad news out of this. We did see that if we let them label overnight, we would get light controlled labeling, but we didn't really see any fast labeling on the fast time scales. And so there was really no difference in our probe versus Matt's probe in terms of the labeling time point. But we went back to the biochemistry a little bit, which was really well characterized by Matt Pratt and his group in his first report of the 6-azid O-GlcNAc probe. And we noticed that the installation of that azide on the 6th position blocks a key phosphorylation site for this biosynthetic pathway toward UDP 6-azid O-GlcNAc. And so that makes the conversion of the 6-azid O probe about 200-fold slower than the standard GLcNAc sugar itself. So we hypothesized we could bypass that slow step by putting in a caged phosphate group. And so Jagaya was able to make that compound in about 7 steps. The key step here is just building up a diphospho or a di-caged phosphine molecule that can undergo alpha selective glycosylation to give this caged diphosphate or this di-caged, PhotoGLcNAc molecule. And we can make this on about 100 mg scale at a time. So it's the chemistry is kind of steppy, but we get enough probe to use for a while out of it. It's inert in cells, so at some of the conditions that we treat at, we don't see strong toxicity. It can decage in minutes. So if we decage for 10 minutes or so, we should see about 70% of it being decaged. And then in our key experiment, when Courtney took this compound and then did her light-based decaging, she was able to luckily see a very strong labeling immediately after we took the sample out and started to analyze by click chemistry. What was very interesting to us is that we also see kind of the second wave of GLcNAcylation occurring about 2 hours after it is cleaved off in about 30 minutes. And these probes, the 6-azid O-GlcNAc modifications, are subject to O-GlcNAcase-based treatment. So we feel that this is being removed and then kind of comes back. And we were interested in probing what is actually happening during these kind of early versus late O-GlcNAc responses. And Suzanne Walker's paper characterizes 2 different time points. They also look at kind of a short versus a 2-hour medium time frame. And they find a very fast buffering response, they call it, where this nonsense-mediated mRNA decay pathway can very quickly buffer out changes in O-GlcNAc homeostasis. But certain factors are probably triggered to program a more lasting change in the cells. And our hypothesis is that some of these aberrant reprogramming phases could be tumorigenic in cancer, especially those driven by hyperglycemia. So we're interested to use proteomics to profile whether any of these splicing factors have key O-GlcNAc sites that regulate either these buffering responses or reprogramming responses in response to our burst of releasing O-GlcNAc. Courtney did some optimization experiments, and she actually finds that the labeling is strongest within about 30 minutes, or within about 15 minutes, sorry. And then that starts to decay and then come back. And it actually seems to oscillate. So we're curious about these types of behaviors. And we think that splicing is at play, at least for some of this. And then she's determined that the optimal concentration is about 300 micromolar, which about matches what the Pratt group has reported for the 6-azido-GlcNAc compound, because if we release 70% of our probe or so, then that matches the 200 micromolar that they usually use for their labeling experiments. I can briefly talk about a proximity labeling system, because again, this is the perfect audience to share this stuff with, even though it's a little bit early. So we're interested in developing an intracellular tagging system that would not rely on us using exogenous levels of sugars. And so Laura Mahal's group published a very cool intracellular biosensor for O-GlcNAc events some time ago that uses a fret pair connected to a GlcNAc binding lectin domain, that when a internal site gets O-GlcNAcylated, that can come together, and then we observe fluorescence indicating that O-GlcNAcylation has been affected at that site. So we took this idea of putting an internal GlcNAc binding lectin with a proximity labeling system developed by Alice Ting called TurboID to put forth these fusion constructs that we call GlycoID. So this is a lectin fused to a promiscuous biotinolase that can bind O-GlcNAc sites within cells and then label those for proteomics. And we could use these to compare cell conditions, for instance. We began by taking the same lectin that Laura Mahal reported. It's a bacterial lectin called GAFD, and putting this on a TurboID system. We indeed see a strong difference in untargeted versus GlcNAc-targeted labeling profiles. And we can do a bit of a time course, although it does take a couple hours to efficiently label. Where we've taken this further, because the affinity of the GAFD lectin for GlcNAc is pretty low, it's about 100 micromolar or so. We've taken this on to a mutant form of human O-GlcNAcase, which binds in order or two magnitude better to O-GlcNAc sites, to ask what sort of proteins are we starting to enrich with these. And so along the way, O-GlcNAcase is kind of a strange protein, in that it can be cleaved during certain signaling events into two pieces, an N-terminal domain and a C-terminal domain, but those two remain tightly associated and their activity is maintained. And so to avoid expressing a long, almost 1,000 amino acid construct with a proximity labeling protein attached, we split it in two, and we'll put our TurboID labeling domain on the catalytic domain of O-GlcNAcase. And then we express the rest of the protein as just a C-terminal unit that's able to stabilize that and help out with some of the binding and labeling activity. And so compared to just our untargeted biotinolase on our left-hand lane here, if we do our co-expression experiment, we indeed see much stronger labeling, and importantly, they'll overlap with an O-GlcNAc labeling antibody pretty nicely. We see some nice dual fluorescent bands there. Okay, so I hope I've shared some tools for observing fast O-GlcNAc events as potentially a new broad regulatory motif used by cells to adapt to their environments. And so we find that some of these fast O-GlcNAcylation events can occur before OGT and OGA levels rebalance in homeostasis. And we hypothesize that these are allowing cells to adapt to different conditions. And like much of biology, they can become broken in disease. We've built some tools to enable time control over these O-GlcNAc events, and we're very interested in probing roles of hyperglycemia, say for in obesity or diabetes, on oncogenic events. And to do this, we're studying some different oncogenic pathways with a professor of oncology at Wayne State. We find some interesting high and low glucose-based effects with some of the inhibitors for the O-GlcNAc transferase or O-GlcNAc-ACE. And we're interested in profiling some of these splicing factors because this TARD-BP protein is a key splicing factor that can be activated in stem cell reprogramming. So the only thing left to do is thank my group, and I've highlighted data presented by Courtney, Sahid, Jagaya, Eamon, and our collaborator, Alicia Bullock-Fisher. So thanks so much for your attendance, and I would love to answer any questions that you have.